Professor Michelle Power is from the School of Natural Sciences at Macquarie University. She is passionate about science and making a difference to wildlife health. Michelle's research focuses on transmission of disease agents at the wildlife-human interface. She studies a broad range of pathogens in different wildlife species. More recently, Michelle's research efforts have focused on examining the ecology of antimicrobial resistance in wildlife, where her group is bolstering knowledge of the wildlife arm of the One Health approach to combating antimicrobial resistance. Thank you, Michelle.
All right. Hello, everyone. I feel like I have to do some comedy to keep up with the others. Okay. You can search my comedy online, actually. I do a bit of science comedy on the side. All right. Look, I just want to say, firstly, I might just acknowledge the lands that we're on today that have been cared for by the First Nations people of this area, and I should have looked it up, I apologise, but also the Indigenous lands that I work on across the different countries that we do our work in. And when I say countries, I mean places in Australia. And also acknowledge any Aboriginal or Torres Strait Islanders who are here in the room with us today, or who are on Zoom out there. All right. So also, I just want to say, look, what a fantastic conference this has been for me. It's a little bit outside of my conference circle. I usually find myself at parasitology conferences listening to stories about poo. I'm going to talk about poo though. And I really want to say, look, the diversity of people that you've got in this audience, and after listening to the sessions yesterday, and you know, some of the complexities that you folks face relative to the global challenges that are happening, that are rolling down to what you as rehabilitators want to do as an individual, you know, care for those animals, release those animals, is really challenging for you all. You know, it's kind of impacting the job and what you really want to do. But around you, you've got this wealth of people and experts. You've got a bunch of young vets here and a few more who are just so engaged in this. And, you know, they're the vets of tomorrow who are going to be the warriors for wildlife. You've got vets who have left that ground room and ground floor and gone into policy and practice through Wildlife Health Australia and through government positions. And those people are influencing from the other angle. So it's really important to realise that that community is out there. And if you can keep that momentum, you can really form a really strong community of practice for when it's needed in coming, well, I was going to say coming months, but that's a bit scary. So let's just say coming. All right, so that said, also going back to another talker, this fantastic photo here is from Doug, and I get to hear his voice messages, so I think he says his surname as Michelle.
Change the cat
okay, I can play it if I had my phone up here. So Doug is fantastic. Doug has supported my research and helped me get into articles and get what we're doing out there, and does a lot of photography and even comes to the lab and photographs what we're doing and in the field. So a plug for Doug. All right, okay. So the other thing is the work and topics that I'm about to talk about I couldn't have done on my own, or my group couldn't have done. And so the plethora of wildlife carers, mainly from South Australia and New South Wales, but also Victoria as well, actually, who have contributed to samples and also data. And just important to say that the integrity of that data and those samples that come in are really important for the science and the evidence we produce to inform change and improve our practice, and really improve the outcomes for you folks as for wildlife, and getting those wildlife back into where they belong after you've done all the hard work rehabilitating them. So lots of people there. I'm going to talk about a few different species and also lots of money that have helped us through. Okay, so before I get into the science side, I'm going to just do a little bit of a background kind of knowledge component. Many of you will probably know some of this, but it will just help us all get through what I would like to talk about. So firstly, antibiotics. Who's heard of an antibiotic? All right, who administers antibiotics? Okay, who prescribes antibiotics? Yep, okay, cool. All right, so we've got a bit of a mix. I'm sure a few more might be also interacting with antibiotics. Who takes antibiotics? Yep, okay, occasionally we might. Cool. All right, so antibiotics, hopefully you all know, are chemicals that kill bacteria. Occasionally they will kill or kill or inactivate bacteria. Occasionally, they will be effective against some protozoan parasites, but interestingly, that's because of an interaction that happened many thousands of years ago between a parasite and a bacteria. So when they do impact or parasites, it's actually because it's targeting the bacterial component that are in those parasites. We can talk about evolution over a cuppa if you're interested. So some that you might be familiar with and that you might have used, Clavulox there and enrofloxacin in wildlife. So antibiotic resistance is really the ability that bacteria have evolved to counter the impacts or the threat or risks that antibiotics pose to them. So really, they've evolved different ways to overcome the action of those antibiotics and manage to survive through when they encounter those antibiotics. So antibiotic resistance is not part of the animal or the person. They're not resistant, and it's not an immune response within an individual or an animal. It's actually the bacteria that have that resistant trait, okay? And interestingly, antibiotics and bacteria have been acquainted for many millions of years, but humans came along, and I always have to think, here's a joke, I always have to think of Alexander Fleming and not Ian Fleming, the Bond author. When I say this, I have to do a double take in my head. But when Fleming discovered the power of antibiotics, and then we discovered the power of antibiotics to treat infections, as prophylaxis in animal production, all sorts of things. You know, we flooded the environment with the different antibiotics that we discovered and synthesized. What we did though in that time is we placed a huge evolutionary selection pressure on bacteria who already had the ability, the genetic ability and the mechanisms to overcome those drugs. So we can see here in this picture, we've got a bunch of little yellow cells, and in the middle there's two red cells, and they're resistant to the antibiotic. So they encounter the antibiotic. The antibiotic kills off all the susceptible cells that don't have that resistance trait or those resistance genes. And eventually you go through and you end up with a population of resistant bacteria, and all the good bacteria are knocked out. And we continue to do this. Well, what have I got there? So antibiotic use continues to do this. So it continues to drive selection for those resistant bacteria that are pretty much everywhere now. And just to give you a bit of a showcase of this, you know, there's a bunch of different antibiotics up there, and you can see on the top row when they were deployed, and then on the bottom row when resistance emerged. And just as an example there, one that you might be familiar with, chloramphenicol, released around 1945, 1946, and resistance was detected, you know, 15 years later, and that varies. Now it can be much quicker, and that's because we've got limited classes of antibiotics. So there's a global undertaking to reduce the risk of emergence of an uncontrollable strain of bacteria through multi-drug resistance. And in Australia, we have this national antimicrobial resistance strategy that guides the use and prescription of antimicrobials, both in the human and veterinary medicine world and agricultural world, anywhere where antibiotics are used. And there's a list of different aims there, but I won't go through those. But interestingly, antibiotic resistance knows no boundaries, okay? For a long time, it was a human issue, but really our continued use of antibiotics has placed that huge selection pressure that antibiotic resistance has just spread through the environment, through our companion animals, domestic animals, and into wildlife. And that's what I'm going to talk about. Why is this happening? We've heard about One Health earlier in the conference, and literally everything's connected. We all share the same environment, as do the bacteria that we carry, that all different animals carry, okay? We pass those in our feces, in our urine, on our hands, when we come in contact with different things. Every bacteria that we carry is potentially being passed in another way. So there's this One Health concept where humans, domestic animals, people, animals, and the environment are all connected, and as are the pathogens. So there's a number of drivers that really influence pathogen transmission, including antimicrobial resistance. And so you can see here, and I guess I'll just talk about the wildlife ones, but you know, one of the biggest things is thinking about, you know, what when we do translocation, either in conservation medicine, conservation practice or rehabilitation, we're taking animals from one location to another. We're also taking the microorganisms that they carry. And some of that has been factored into some of the the animal management codes and conservation codes. All right, so something else I wanted to highlight is we hear about zoonoses a lot. I wanted to highlight reverse zoonoses, okay? And this is actually where human-associated disease-causing agents are spread to wildlife. And really one of the biggest examples of that is antimicrobial resistant bacteria. And just the last little, I think this is the last piece of knowledge we need to have in going into the science part, well, I guess this is the science part, but is that, you know, we know about, or you might have heard about, you know, severe pathogens that are antibiotic resistant. So a Vibrio cholerae, so the bacteria that causes cholera, or tuberculosis, okay? But the biggest problem or concern is things that were once not pathogenic or didn't have the ability to cause disease that now do, and are also antibiotic resistant, antimicrobial resistant. And these are things like E. coli, Klebsiella, Salmonella, Enterococcus, things that you folks might be aware of or have encountered through wildlife care. Okay, so we've been investigating for probably for a good 20 years now, in trying to just get, in all that time, trying to just get some baseline data on the spread of antimicrobial resistance to different wildlife species. And now we're going forward with some really applied questions and hopefully looking at ways the work we've been doing can translate into practice and prevent detrimental outcomes through spread of resistant bacteria. So we've been looking at animals in marine systems, terrestrial systems, and also in conservation programs. I'm going to touch on some of that data today. Each of them has their own story, but in the 23 minutes I've got left, not enough time to cover all that. Okay, and I just want to tell you a bit about the lab work that we do and how we test for antibiotic resistance. Pretty much start with a fecal sample or a rectal swab or a swab of some description, and then we extract the DNA. We go through and do genetic tests. So we can test for mechanisms of resistance, we can test for specific resistance genes to certain antibiotics, and we can also culture the bacteria and do whole genome sequencing, and also sensitivity tests where we expose those bacterial isolates that we've cultured to different antibiotics and see if that antibiotic works or not. Okay, but the one particular element that we started on is called the class 1 integron, and we chose this because it's a type of mechanism that's been involved with antibiotic resistance in enteric bacteria, so your E. coli, your Klebsiella, okay, and it was popping up in different species around the world, so as in animal species or host species. So just a little bit of simple knowledge here, I hope. We can see bacteria here, it's got an umbrella. That umbrella is red and green. And so when the bacteria is exposed to the red and green antibiotic, it's able to resist it, okay? It doesn't have any impact. It's got some mechanism that takes that away. Down the bottom is just kind of a little schematic of the genetic mechanism. Bacteria gets exposed to a new antibiotic, but this particular mechanism here has the ability to sequester or capture resistance genes in the environment. So resistance genes can float freely, and just through genetic or DNA processes, those resistance genes can be captured by bacteria and then give them that selective advantage when they're exposed to them. So very quickly, this can happen and a bacteria can come resistant. And then basically can just keep going. We've got this little resistance cassette can actually hold up to 10 different antibiotic resistance genes. And so you've got multi-drug resistance in a bacteria that just happens to have this, okay? So the genes can be sequestered in and out of this little capture system, I guess is a simple way to explain it. But just gets a little bit more complicated is the bacteria can actually share that little capture system with other bacteria of the same species or in different species, okay? And this is why this particular mechanism, the class 1 integron, has been particularly involved in spreading resistance between different bacterial species within the enteric system. Okay, so this just shows those genetic mechanisms being spread between different bacteria. And I should say, because it's going to be important later, that kind of sharing of resistance genes happens when a bacterial community is under stress from exposure to antibiotic or under stress from anything that might incur or induce stress in a microbial system. All right, so this is kind of a bit of an important slide. This captures really the work we've been doing over the years on different species, just looking at the occurrence of class 1 integrons, so that mechanism that can carry those antibiotic resistance genes in different wildlife. And we've compared between animals sampled in free-range habitats, so in the grey bars as wild, or in captivity in some description. And it varies, it depends on the species. Some might be long-term collections in a zoo, others might be rehabilitation animals that have come in for various reasons. So it depends on the species relative to why they have that connection to humans. But really the thing to note is across all species bar one, when animals come in closer to contact to humans or in care or captivity for some reason, they have a prior propensity to carry antibiotic resistance genes. Okay. In fact, they actually acquire it when they come into those environments, and we'll see that through some of the examples, compared to the free range animals. Interesting to note, though, when you also see animals that are have that high connectivity to people, such as the possums, in wild environments, you also see quite a high carriage. And the Tassie devils are an outlier, and someone might ask a question at the end about that. Okay, so I just want to get into, so maybe what I'll say is, so what we can do is we can go in here, we can look using very simple tests to see if these resistant mechanisms are there. And then once we find them, we can go and sequence and work out what resistance genes are present, and then what that might mean for use of antimicrobials in these species or in wildlife generally. Okay, so one of our focus species first is the grey-headed flying fox. And I'll just get straight into the data. So the first slide here shows the different bacterial species that we've actually isolated from grey-headed flying fox species. So we've got a sample, we've gone in, we've done culture, and we've targeted either E. coli or Klebsiella or Salmonella. We've compared this between adults in green and in pups, okay? We can see generally that in pups, most of those are in care. In pups that we actually see a high prevalence of E. coli, and many of these E. coli are resistant to antibiotics. So some to the beta-lactam class, some have multi-drug resistance, and then there's certain types, because we can do sequencing and work out what strains we're carrying, we've got certain types of E. coli. So extra intestinal bacterial strains which cause infections and outside of the enteric system, okay? But basically, the important thing to note is these are all pathogens for people. Many of them have been associated with, are more associated with humans than they are with wildlife. Then we've got some pathogenic E. coli, pathogenic Klebsiella and antimicrobial resistant Klebsiella, and down the bottom we've got some Salmonella. So resistant bacteria out there, many of these strains are pathogens, cause infections in a wide range of species, okay? And these are just sitting quietly in these animals and at this point in time, not causing any harm. So if we look at some of the stats, I might just, maybe I'll grab this. Are we on? Are we on? Can we just grab this one? Thank you. Yes, no, yes, cool. Okay, so we can see that 50.9% of pups carried multi-drug resistant isolates, okay? And this was to beta-lactam antibiotics, ampicillin, amoxicillin, clavulanic acid, cefalaxin and cefotaxime, and I'm probably not pronouncing them right. So, but really just important, like the message I want you folks to take home is that there is just a huge amount of antimicrobial resistant bacteria out there that you just don't see and that you don't know about until those antibiotics come along and perhaps generate a risk for that animal. All right, I'm going to go back to you. I can't do the handheld, sorry guys, I can't do the handheld one. Okay, so more importantly, when we have when we have emergencies or major impacts to animals, we do see a shift in those antimicrobial resistance dynamics, okay? So this is a heat stress event, and we can see animals sampled in 2018 prior to a major heat stress event in 2019. And what you can see, there's just a couple of antimicrobial resistant strains there, those green ones. But then when you look across that heat stress event, you see this huge diversity in the different types or strains of bacteria, and also the antimicrobial resistance prevalence changes. So why we're still trying to delve that out, a lot of it is probably because these animals are coming in, going into small enclosures, encountering other animals, and things are being reinfected from the environment and so on. But we're still trying to tackle what drives this. And then the other thing is I said we can look at the genes to the different drugs that they're resistant to. And if we look at this slide, it's breaking down to 4 antibiotic classes. So we've got the beta-lactams up there, the chloramphenicol, tetracyclines, actually more than 4, fluoroquinolones grouped together, the aminoglycosides, and the sulfonamides and trimethoprim. And the different coloured bars are just whether we analyse these from bacterial culture or whether we use direct gene detection. But just to note the numbers down the side here, you know, we've got beta-lactam, 33 different antibiotic resistance genes in these flying foxes. So beta-lactams, 11 different ones, aminoglycoside resistance genes, 7, chloramphenicol, 2, tetracycline 2, and to the fluoroquinolones. And so from both veterinary and human medicine, the fluoroquinolones are one of the last lines of defence, okay? That's enrofloxacin. So if we start to see the frequency of some of these key drugs out there, we start to get bigger problems than we've already got. And then finally, the sulfonamides. Now, I did change my slides a bit and added some of these numbers to try and make it a bit easier. And even last night when I added this up, I'd never really gotten into my head that it was 33 different genes just in flying foxes alone. And seriously, it actually scares me. All right, one of Doug's photos. Okay, so moving on to koalas. Now, I guess this is where the occurrence of antibiotic resistance is kind of critical, because with chlamydia in koalas, the only current defence against that is the use of antibiotics, and the 2 main ones being chloramphenicol and doxytetracycline. So we had a Morris Animal Foundation grant that was in partnership with WIRES and also the Koala Conservation Australia, and we were looking at a number of different things in this project, and I have circulated the survey cards, and we will send out a link, and I'll give a plug about that at the end. We've used treatment records to look at what antibiotics were used in that emergency response and when animals went into care. But then finally, we also want to understand what happens with those antimicrobial resistance dynamics with koalas when they're being treated in care. So we had a number of collaborators on this project that made it possible, and some are here, over there. Okay, so what we really wanted to do was to investigate the antibiotic resistance ecology in fire-affected koalas, and another of Doug's photos. And if we look at this chart, we can see that really we go out, we actually do have low-level background resistance of presence of resistance genes to those antibiotics. And here we've got, it doesn't say, oh, sorry, no, this isn't, this is the integron frequency. Sorry, this is, just take a step back. So this is that mechanism that I've already spoken about, okay? So here we look at pre-fire samples. There's no significant difference between the pre-fire samples and post-fire samples in Mount Lofty Ranges for the presence of those class 1 integrons. But on Kangaroo Island, there was a significant difference. And for some reason, the animals, there was no antibiotic resistant class 1 integrons present in animals sampled on Kangaroo Island before the fires across 2014 to 2017. But you see we get up to close to 50%. In animals post-fire. Okay, The other interesting thing when you break that down is you look at those samples. We did have animals from the wild there, so we do see low level just across the board with all the samples that we had. But when animals again go into care, we see that increase, and really just showing that for some reason there's something that's happening. And I'm not necessarily saying that it's use of antibiotics in this instance, but there's something that's happening that's shifting the dynamics and driving that sharing or emergence or acquisition of resistance genes. Okay, so then we detected different genes to different classes, so the aminoglycosides and trimethoprim. So, and some of these antibiotics are used in clinical medicine. So what I just want to point out is there with the integrons, we see trends pre and post fire and shifts and increases. And similarly, like with the heat stress event, in emergency events, there is something that's driving a change in antimicrobial resistance dynamics. So what we can do with our work is we can actually look for the specific genes that target or enable bacteria to resist certain antibiotics. So in this instance, we were looking for resistance to chloramphenicol and the tetracycline, so the two antibiotics used to treat chlamydia. So when we look just in background samples, we can actually see that those resistance genes are present in koalas at low levels, okay? And so we've got CAT, which are the to chloramphenicol resistance genes, and then 6 to tetracycline. So we're picking those up as background in koalas. When you actually look at the frequency of those genes in koalas that had been tested for chlamydia, we can see here in this table, so there were 22 animals, low numbers, we're still doing this research, 11 negative and 11 positive, okay? So we can see here that in a positive, chlamydial positive animal, we've got CAT there in 2 koalas, sorry. In positive again, CAT A2, the second type of gene in 2, and just the trends down there. But what's also important to note is that we actually have individuals carrying resistance genes to tetracycline and tetracycline and chloramphenicol. But equally, we've also got animals that were negative for chlamydia that also are carrying those genes, and just highlighting that those resistance genes can be present in animals that haven't necessarily had antibiotics or are exposed to antibiotics. All right, and so again, looking at shifts in what happens with those two particular genes relative to a fire emergency. So really just the general trend there, pre-fire, we have low prevalence. So here, less than 2% really, but then it shoots up to about 8%, low level at the moment, but still. So increased prevalence, but then we also have increased diversity, and you can just see in the coloured bars, this is pre-fire, fire affected, that we've got all pink, but we've got 4 different types there, 2 here, and 4 there. So really, again, just showing those shifts in the dynamics. And that's really just what I'd like to highlight with the data, is we are seeing these shifts again relative to emergencies. So what does this mean for the animal? Okay, I've mentioned that if resistance genes are there, the exposure of bacteria that are resistant to an antibiotic will then select for them. So you can get amplification of resistant strains. Some of those resistant strains may be pathogens and then may lead to infection, wound infections, in systemic infections, and then detrimental outcomes for the animals that you're rehabilitating. I mentioned that the dynamics of antimicrobial resistance sharing between bacteria is actually influenced by stress. And by this, I mean microbiome stress. So when an animal does receive an antibiotic, imagine all the bacteria in there saying, oh my God, like you've just given me something that's going to wipe me out, okay? So they all go into overdrive and literally just start grabbing things from the environment to see if they can get a gene. So you can potentially have amplification and sharing of those resistance traits within that microbiome, and possibly to endemic, which is something we've never looked at, possibly to endemic bacteria that should be there, the bacteria that are naturally part of the animal's microbiome. So really the implications are secondary bacterial infections, increased risk of disease and poor health outcomes, and then risks for the overall outcome of that animal being released. Just coming back to this picture earlier, and I'm just using it just to highlight the transition to the next one, where just showing that it's actually across species. We don't just have one gene there again. We've got multiple resistance genes. Some animals carrying multiple resistance genes, others not, to different classes of antibiotics. And really kind of, I think we're at a tipping point just generally around the world for the emergence of resistant bacteria. So in different animals, we have multiple resistance types. And so what that means is that we've had multiple acquisitions of resistant bacteria over time. And if we just take a look here, this is just a subsample. It's a bit of an old slide, this one. But again, to 4 classes across our 6 species over there, the echidna, platypus, possum. And this is just looking at the different resistance genes that are present across those species. But basically, you know, a total of 395 total detections of a resistance gene in around 1,700 animals that were primarily in rehabilitation. Okay, so really important to start thinking about what this means for your practice and use of antimicrobials and antimicrobial stewardship. So the implications are that there's treatment implications in wildlife hospitals and rehabilitation centres. There's a consideration of antibiotic stewardship during emergency responses. There's resistant bacteria. You know, these represent invasive organisms. They shouldn't be there. They're colonising those wildlife microbiomes and shifting the important dynamics of that natural system. So there can be microbial level disturbance that we just have no idea about or how that might impact the health of those animals. And then resistant strains are also typically pathogenic. And so that means you can have those detrimental outcomes or infections that you just don't know where they've come from because they've gotten an antibiotic. Okay, by looking at this, I think I've got about 3 minutes left. 5 minutes. Okay, I've got 5 minutes. Okay, cool. Okay, all right. So just to sum that up and then maybe to give you some actions to think about, and I am asking for more change. So, you know, we've got a healthy animal in the environment carrying a resistant strain. Maybe in the natural environment where everything's good for the animal, there's lots of feed, there's nothing else disturbing its health, that resistant strain of bacteria just hangs out, okay? Something might happen, shift those dynamics, or the animal comes in for some other reason, and then in care, and then they may need some treatment. And it's at this point then that treatment might influence those resistant bacteria that are present in the wildlife microbiomes. Then, you know, happy outcomes, that animal's released, but they're also potentially taking those resistant bacteria with them and into the environment and to their counterparts back in their free-range environments. So really important to think about, you know, biosecurity and pathogen spread in those actions as well, and what that might mean for the population if a resistant organism is spread out into a wild population. Okay, so some recommendations, and I'm sorry, I did these last night. I did hand my slides in a long time ago, and I'm a night before writer, so you don't have these, but I'm happy to be contacted and I can give you these as well. Okay, so really these are some general principles around antimicrobial stewardship. Some of them apply to vets because they're prescribers. Some of them apply to vets and anyone working with wildlife and rehabilitators, and others to rehabilitators. Okay, so I'm sorry, I feel like I'm ignoring this side of the room. All right, okay, so you know, the first thing is prescribe antibiotics only when necessary, and that's for the vets, okay? They are actually under new guidelines influenced by that Australian National Antimicrobial Resistance Strategy to not prescribe antibiotics when there's no evidence of an infection or when they're not needed. So we have had in the past, you know, we'd go to the doctor and we're sick and the doctor would always, I remember when I was little, you'd always walk home with a prescription. That can't happen anymore, both in human medicine or veterinary medicine. So going in or having an expectation that you might take an animal to a veterinarian and you might leave with antibiotics because maybe you've had them before, is just that expectation has to change, because the vets themselves are under someone can give me the right words here. Under what was it, Tamsin? Under scrutiny, there we go. I didn't. Yeah, so they're under scrutiny from the people from the AVA and people who manage them, but also influenced by guidelines. Okay, so really important to keep that in mind. They're not giving you the antibiotic because they don't want to, or because they don't think it's needed, or they don't care. They're not giving it to you because it's not needed, because there's no infection, and because they're under stringent conditions and rules from managing bodies. When vets do prescribe an antibiotic, they will choose an optimal antibiotic for that infection or for what they've diagnosed, okay? They will provide an optimal dose and also a time period, and that has to be followed. Changing those shortening times, prescription times, delivery times, manipulating what the recommendation or the information is from the vet actually is against good antimicrobial stewardship and drives the emergence of resistant bacteria because they're just not exposed to enough drug. They're not cleared. So treat as correct doses and complete the course as prescribed. Consult a veterinarian before any use of antibiotics, okay? If you've got some, you shouldn't have some left over because you will have given the animal what they needed. But, if you've got some left over for whatever reason, you shouldn't be making decisions to give a little bit or give some to an animal this day and something else the next day. And I hate to be harsh, we know this is happening. We see this on their Facebook pages, okay? We see the suggestions, and maybe not here, but I'm not sure where our Facebook pages over in New South Wales spread to, but we see, we tried enrofloxacin one day and then we tried something else the next, or we gave it 1/2 ml, then we gave it 2 mils. It might have worked for an animal one time, but again, how many animals did that not work for? Okay. I've mentioned there, recognise that vets are being educated on good antimicrobial stewardship, and I've flagged that there. Adhere, this is getting into, there's a lot of recommendations out there. Actually, I'll get to the next one. This is better. I'm getting pressure. This is getting red. Look, there's a lot of information out there that you can now source as wildlife carers and also for the veterinarians in the room. We've got the National Wildlife Health Biosecurity Guidelines, which cover antimicrobial resistance, but also the general practice there around hygiene and biosecurity actions to reduce transfer of our organisms to your charges will help inform you. There's fact sheets on antimicrobial resistance at the WHA site. There's various recorded talks. The treatment care and guidelines, at least in New South Wales, and the code of practice for different species are now drawing on the evidence-based research around antibiotic resistance in wildlife and making recommendations around treatment and use of antibiotics, and really giving some of these as an examples of what is good practice. And then there's also the Medicine of Australian Animals. Some of you may have access to that. That's got information there too. And then the last thing I just want to plug is we really do want to get an understanding of what's happening around antimicrobial resistance, your knowledge, your practice. And then the next steps for us is to really move and shift to developing some training workshops and some educational resources to help you get through this. I think I'm over time and there's probably no time for questions.
Yeah, so one of the things when we were putting together the agenda for this conference, we did make a bit of a conscious effort to shy away from research papers, given that this is a wildlife rehabilitators conference, and we try and keep it relevant to wildlife rehabilitators. But I think we can all agree, I hope, that was a very informative presentation and extremely relevant to, I think, every single person in this room. And it's even given, all of us at our table having some chats, a lot to think about, for our own hospital and things. So thank you very much. Really